Good afternoon or good day. Welcome to our seminar this afternoon. It's the first MHRC seminar of the semester. And I'm very pleased to introduce our speaker today, Dr. Joe Gordon, who's at the University of Manitoba. I'll tell you a little bit about Joe's background. Joe is originally from Newmarket, Ontario. He did his undergraduate degree at McMaster University. And then for his master's, he came to York University, and I was very lucky to be able to work with Joe for his master's degree. We did some nice work on mitochondria and protein import in the early, or about the year 2000 or so, published some papers in 2001. Then Joe took some time off. He worked at Seneca College and spent some time teaching there. For about 4 years, and then came back initially part-time to do his PhD back here at York with Dr. John McDermott, where they studied MEF2 signaling in smooth muscle and the role of that transcription factor. It was very productive, and then did a postdoc at the University of Manitoba with Laurie Kirschenbaum, where he got back to studying mitochondria and cell death, and he's been there at the University of Manitoba ever since that time, now as a professor in the Department of Nursing and the Department of Human Anatomy and Cell Science. So today, we're going to be pleased to hear him talk about molecular regulation of muscle mitophagy. So that'll be exciting. Joe, if you don't mind then, please turn on your screen and unmute yourself, share your slides with us, and we can take it away.
All right. Well, thank you very much for the invitation, Dave. Today I'm going to talk about the molecular regulation of muscle mitophagy, and my subtitle is a nexus of mitochondrial quality control and cell signaling modulation. And you might wonder why nexus, but I'm using that term as a convergence, or bringing together these two seemingly opposite or disparate processes into one kind of regulated piece. And so I'm excited to share some published data today and some unpublished and some even very preliminary data. As Dave mentioned, I'm at Children's Hospital Research Institute of Manitoba, and I'm a member of the DREAM Diabetes Research Team. And DREAM exists because Manitoba has the highest incidence of youth onset type 2 diabetes in Canada. And so we brought together this team, and this is our mission and what we do. And I'm a basic scientist within DREAM, and so we use cell culture and animal models to investigate aspects of muscle insulin resistance, and then some diabetic cardiomyopathy and remodeling, but I'm not going to show that today. So before I begin, I'll do a land acknowledgement. The University of Manitoba is on Treaty 1 territory, and this is the original lands of the Anishinaabe, Cree, Dakota, and Dene peoples, and the homeland of the Métis Nation. And we respect the treaties that were made in these territories. We acknowledge the harms of the past, and we move together in the spirit of reconciliation. So when I started my lab and joined DREAM in 2012, we became really focused on this question. What are the mechanisms that link mitochondrial dysfunction with insulin resistance in muscle? And let's talk about mitochondrial dysfunction for a minute. It's long been noted in human studies and animal models that there's a form of mitochondrial dysfunction in muscle, but it's fairly ill-defined. We don't know a lot about it. It has been proposed to be a form of overload, where the mitochondria get overloaded by too much fat. And this is a process called lipotoxicity. And there's accumulation of fat and intermediates in the muscle, which has traditionally been known as steatosis. And so these overloaded mitochondria are somehow sending signals out, and we don't know a lot about them. We know that some protein kinase C isoforms are involved, but there are cell signaling events that impair muscle insulin signaling. And some of the best known targets are the insulin receptor substrate 1, and it's inhibited, and this reduces insulin-stimulated glucose uptake. And one of the key features or the earliest findings with this mitochondrial dysfunction was a reduction in the mitochondrial biogenesis regulator, PGC1 alpha. Some of the other things that we knew at the time were that exercise training, and in fact, even an acute bout of exercise, can resensitize muscle for up to 72 hours. And when that happens, we know that this steatosis changes. We get changing of lipid intermediates, we get more triglyceride storage, mitochondrial function improves, and whatever brake that or molecular brake that's been placed on the insulin signaling pathway is restored, and we see improved insulin-stimulated glucose uptake. And John McGavick and Vern Dolinsky and I wrote a review early on in that course where these images came from, and I have to thank my brother for drawing these and helping us publish that review. One of the things that caught my interest early in the process of studying insulin signaling and insulin resistance in muscle was the process of autophagy. And there's quite a famous paper published in Nature by Beth Levine's group and Joe Hill at Texas Southwestern, showing that the autophagy response actually helped sensitize muscle to insulin. And this was a really novel observation, but it was quite perplexing to me. And the more I thought about it, the more I couldn't let it go. And the reason is autophagy was originally described as the cellular response to starvation. And in muscle insulin resistance, we're in an overfed state. And I couldn't reconcile this observation with how is autophagy regulating insulin sensitivity in an overfed state when it should be inhibited. And if you're not familiar with autophagy, essentially in a typical starvation scenario, we get the initiation of the process and the formation of an autophagosome is formed. And this double membrane structure essentially sequesters dysfunctional organelles, misfolded proteins, and delivers these cellular material or cargo to the lysosome. And the lysosome digests it and liberates nutrients that would allow the cell to survive this starvation response. And when we were thinking about this, that's neat and all, but what's going on, and does this relate to mitochondrial dysfunction? And there was a lot of questions that started arising when we started kind of questioning how autophagy might be involved. So when we looked into the literature, another observation that really caught my interest was that mitochondria become fissioned during muscle insulin resistance. And fissioning is essentially the aspect of taking a large mitochondria, breaking off smaller bits, which then can be recycled by the autophagy response. And a lot of this breaking apart and fissioning of mitochondria is regulated by the dynamin-related protein 1. Which takes these fissioned mitochondria and, like I said, delivers them to the autophagosome and lysosome. And then the remaining mitochondria, which is presumably functional, fuses with other functional mitochondrial bits, mostly through the regulation of mitofusins and OPA1 to kind of complete the cycle where we have a restored mitochondria. And the process of mitochondrial biogenesis feeds into this. But since we were interested in lipids and lipotoxicity, the observation that really caught my interest was that some mitochondrial phospholipids are involved in regulating fission and fusion. And I started wondering, are these mitochondrial phospholipids, are their content or their structure altered by high fat feeding and muscle insulin resistance? So the first the first phospholipid that's been implicated in mitochondrial fusion was cardiolipin. Cardiolipin binds with OPA1 and facilitates intermembrane fusion. However, this cardiolipin can be broken down into phosphatidic acids, and that's the second phospholipid we became interested in. And phosphatidic acids can regulate fusion and can regulate fission depending upon their structure and their content, where saturated versus unsaturated fatty acids can impact phosphatidic acids and how they regulate DRP1. And so with this kind of information, we got the fission process and a lipid process. We started thinking, hey, let's see if these things are related. And my lab was really focused on the time studying two related BCL2 family members called BNIP3 and NIX. NIX is also known as BNIP3L. And these are death genes, but they've also been associated with a mitochondrial autophagy response, what I'm going to call from now on mitophagy. And essentially what they do is they target or elicit mitochondrial depolarization, so the membrane potential is reduced. And then NIX and BNIP3 can interact with what we call ATG8 family members to recruit a growing autophagosome or a phagophore and essentially capture dysfunctional mitochondria and lead them for degradation. And so we started wondering if, hey, is the autophagy response we're seeing in insulin resistant really the mitophagy of fissioned mitochondria? And so we started looking at that. Is that the mitochondrial phenotype in insulin resistance? And then how does that elicit cell signaling pathways? And so working with some collaborators, notably Vern Dolinsky within our DREAM theme, we collected skeletal muscle from either low-fat or high-fat fed. It started as rats, but now we're doing mice. And I do want to come back and talk about these diets a little later on because they're actually a high-fat and sucrose diet and a low-fat and sucrose diet. And that actually is going to be important to my story a little later on. But we saw when we took soleus muscle and we Western blotted for NIX, we saw quite a marked induction of NIX at the protein level in high-fat fed animals compared to low-fat fed animals. And at the same time, we saw concurrent dephosphorylation of DRP1 at a calcineurin phosphorylation site. And so this dephosphorylation event actually activates DRP1. And so we saw these things happening together in vivo and then went about mechanistically trying to determine, are they related? Mechanistically, we wanted to use a cell culture model, and so we use C2C12s either as myoblasts or myotubes, and we also use L6 myotubes because their insulin signaling pathway is more intact. To elicit a lipotoxic response in our culture models, we put the saturated fatty acid palmitate into the media, and we can see a real quite marked NIX induction in this model. And so we're going to use these two model systems together to try and mechanistically tease this out. And I should point out that most of the work that I'm going to talk about right now was published by a PhD student in my lab, Simone de Silva Rosa, who's now a postdoc in Gary Sweeney's lab. And so to kind of get some clues of where we were going, I got into metabolomics. And metabolomics is a mass spectrometry technique. And we started by doing what's known as shotgun metabolomics. It's not truly quantitative, but it can give you an idea about abundance of lots of metabolites. And we, in a single shotgun run, we can get about 12,000 metabolites, and we separated out water soluble and fat soluble metabolites. So 12,000 fat soluble metabolites was kind of what we got and had to work our way through. And we paired this with a gene expression array, mostly for muscle and autophagy related genes. And so we took our high fat and sucrose fed animals, and we can see the triglyceride content was increased by quite a bit, and that was already known. The diacylglyceride content was really quite elevated, and this is one of our key lipotoxic lipid intermediates, as well as ceramide content were quite markedly upregulated or accumulated in insulin-resistant muscle. And again, this is another lipotoxic lipid intermediate. But when we looked at cardiolipins and phosphatidic acids, which were kind of our new kind of ideas that we were throwing around, we see that the most abundant cardiolipin, which has 4 fatty acyl chains, was really downregulated. It wasn't there, and it was replaced actually by cardiolipins that have an altered composition. But what was kind of interesting to me is that we also saw an accumulation of phosphatidic acids, and phosphatidic acids with both saturated chains and unsaturated chains, including the alpha linolenic acid that we would find in cardiolipin was accumulated quite a bit. And this suggested that maybe there's an interplay between these two things. When we looked at our gene expression array, we see lots of autophagy genes. And just as we would expect in a high fat or overfed state, they're downregulated. And so that kind of gave us the idea that macroautophagy in a starvation sense is not activated in insulin resistant muscle. In fact, it's probably inhibited. And we also looked at PGC1 alpha as a kind of a characteristic marker of muscle insulin resistance, and it was downregulated as well. So then we went to our culture model, and I really wanted to study mitophagy and the mitophagy response. But it's not an easy thing to study. And what really changed it for us was a biosensor, the availability of biosensors from the AdGene repository. And the one that we really jumped on was mitofred. And mitofred is a newer version of mitochondrial matrix targeted chyma. It's more stable, it's brighter, and much easier to image. And so we had that available in a plasmid form to us, and we could transfect C2C12s quite easily, and then evaluate mitophagy just by quantifying the red fluorescent signal. So you can see when we treated our C2C12s with palmitate, we could activate the mitophagy response. And when we knocked down NIX with an shRNA, we could blunt that response, and that's quantified here. Just to show you other ways that we can look at mitophagy, one other common way of looking at autophagy in general is looking at the LC3, which is an ATG8 protein fused to GFP. And a lot of times people look at the collection of these of the GFP signal in dots to indicate autophagosome formation. And you can see that shown here. And one of the reasons why I avoided this field for so long is it's not very remarkable, and people spend a lot of time counting GFP dots to try and quantify this process, and just quite frankly had no interest in doing that or trying to convince a student to do it. So we avoided the area for a while, and until we had the adequate tools, we thought, to really dig into this. Another angle we went after was calcium signaling, and we had previously shown that NIX regulates a transfer of calcium from the ER or SR to the mitochondrial matrix, and that's involved in mitochondrial permeability transition. And since DRP1 is activated by calcium, we wanted to know, is NIX actually triggering a calcium response that could lead to that activation? And so we used an ER-targeted calcium biosensor called a GECKO from Robert Campbell's group at the University of Alberta, and a mitochondrial matrix-targeted GECKO and treated cells with palmitate. And you can see the ER calcium content gets depleted with palmitate exposure, and we get a simultaneous increase in the mitochondrial calcium content, and both of these effects are eliminated by NIX knockdown. So that gave us some evidence that NIX might be altering calcium signaling in this process. And then we thought, if the calcium is going to the mitochondria, is that picking up and activating signaling pathways along the way, like activating DRP1? Before we did that, we looked at the mitochondrial morphology, and we just used epifluorescent microscopy with mitochondrial targeted emerald fluorophore. And you can see in C2C12s, when they're healthy and sitting there, they got quite the spaghetti or fused mitochondrial morphology. And when we transfect in NIX, we get a fissioned kind of phenotype, or a really quite robust fragmented phenotype, where the mitochondrial cluster around the nuclei, and previous work suggests that that's a mitophagy kind of pathway, where the mitochondria get pulled towards the nucleus to be degraded by the lysosome. And so we've quantified that here. When we add NIX in, we see a decrease in the elongated and an increase in both intermediate and fragmented mitochondria. When we turned our attention to DRP1 and we transfected NIX in our C2C12 line, we could see a reduction in that calcineurin dephosphorylation site, suggesting this is calcium regulated, and NIX is directly able to activate DRP1 when it's transfected in without really impacting mitofusin or total DRP1. And then we looked at mitofred, and we add NIX, we see an induction of mitophagy. And this can be blocked if we use the fission and DRP1 inhibitor called MDivi1. And as a positive control, we inhibit lysosome function with baflomycin A1, and we can also see that reduction. Finally, the real point of this was, is this mitophagy response impacting insulin signaling and insulin-stimulated glucose uptake? And so for this series of experiments, Simone used a combination of C2, C12s, and L6 cells, which are more coupled for insulin-stimulated glucose uptake. And through a series of Westerns, she showed that when she transfected in NICS, and you can see her transfection below, and then we treated with insulin for 15 minutes, we could actually activate an inhibitory phosphorylation site on IRS1, implicating that insulin signaling might be inhibited. And this phosphorylation site is thought to be a negative feedback inhibition from insulin signaling directly. So you can see it's activated a little bit with insulin, but much more robustly when NICS is present. And this also translated into a reduced insulin-stimulated glucose uptake using the 2-NBDG fluorescent glucose analog. And when we did the opposite experiment and palmitate treated, we could inhibit insulin-stimulated glucose uptake, and then we knock down NICS, we can restore it. So gain of function and loss of function studies showing that NICS can regulate insulin-stimulated glucose uptake in culture, and the likely mechanism is by inhibiting IRS1. So with this in mind, we kind of had the pieces in place, but we didn't really have a mechanism. We knew NICS was involved in mitophagy and fission, and we knew it could inhibit insulin signaling, but we didn't really know how. And this took us quite a while to figure out, and we tried a lot of different pathways that just didn't pan out. And so just through process of elimination, we just kept moving on to the next one. But the pathway that kind of panned out for us and one that caught our interest was the negative feedback pathway. So when we treat cells with insulin, we activate the typical PI3' kinase, AKT, GLUT4 translocation pathway. But there's also known pathways where AKT can activate mTOR, the mammalian target of rapamycin. And through P70S6 kinase, mTOR signaling inhibits IRS1. And that's exactly what we were seeing with NICS. So we wondered if this pathway was involved. And what really caught our interest was that when we looked at mTOR signaling as a potential player in this, we found that mTOR has a requirement for phosphatidic acids as a lipid binding protein or a lipid binding molecule to be activated, and that a large portion of the cellular mTOR composition is located at the lysosome, where it's regulated by Rheb, which is another GTPase binding protein. So this kind of got us excited that maybe all these pieces will fall together. And as we were just beginning this work, a cell metabolism paper came out showing that Rheb and NICS cooperate together to activate mitophagy. So Simone jumped on this fast, and we started investigating. And what she showed was that when she transfects NICS into either C2, C12s or L6 cells, we get an activation determined by phosphorylation of P70S6 kinase. And this is inhibited by rapamycin treatment, which inhibits the mTOR TORC1 complex. She also did the opposite experiment where she palmitate treated and see P70S6 kinase activation, and knocking down NICS prevented that. This process was also blocked by MDV1, which is the mitochondrial fission inhibitor. And if we pre-treated cells with 1-butanol, which prevents phosphatidic acid production, we can also eliminate that response. To just take it one step further, we started knocking down some components of either the Rheb pathway or the cardiolipin to phosphatidic acid pathway. And so again, doing the same type of experiments where Simone overexpressed NICS, she could see P70S6 kinase activation. If she knocked down Rheb, which is thought to be that in-between molecule that activates mTOR, that response is blunted. Or if she knock down mitochondrial phospholipase D, which is phospholipase D6 isoform. And this is the enzyme thought to be responsible for converting cardiolipin to mitochondrial phosphatidic acids. That also eliminated the response. And finally, when she looked at Rheb and NICS-induced mitophagy, this response was blocked if we prevent NICS from getting to the mitochondria. So if we mutated the transmembrane domain of NICS, we lost that mitophagy response. And if we put in a phosphomutant that prevents NICS from translocating to the mitochondria, that response was also lost. So this part of the project is kind of summarized here, and this is our big molecular contribution to the field. It was mostly done in cell culture, and I'll talk about where we're going with it next. But based on the literature, we described kind of what is a classical pathway that during lipotoxicity, diacylglycerols accumulate, and there seems to be a block from converting these to triglycerides. The DGs activate PKC signaling, most notably PKC theta, and that inhibits IRS1. But Simone's work describes an alternative pathway, and that's what we're trying to describe it as, where lipotoxicity also induces NIX expression. NIX can release calcium from the ER-SR to activate DRP1 and recruit lysosomes to dysfunctional mitochondria. And from there, we're going to set off a signaling pathway originating from the lysosome involving Reb and phosphatidic acids, which activates the mTOR S6 kinase pathway to also inhibit IRS1. And so this is kind of where we are with our lipotoxicity pathway. And I'm going to come back to this model in a minute, but I'm going to show you a concurrent project that we had going on in the lab first. And that involves taking C2C12s, differentiating them to the point that they become contractile myotubes, and then pacing them electrically, even for just an hour at 1 Hz. And we're going to use this as a model of a real reductionist model, if you will, of exercise in a dish. And this is just some pictures of what they look like. You can see by day 10, we get really quite fat, robust, and contractile myotubes. And this is them with MF20 staining. So when we started pacing these myotubes, we kind of saw, well, what you would anticipate, the opposite to happen to PGC1 alpha, the mitochondrial biogenesis regulator. Its expression goes up. But we also saw NIX expression going up at the same time. And that kind of got us thinking that, hey, maybe NIX is regulating mitophagy in lots of different muscle stresses. And maybe what PGC1 is doing kind of dictates whether we have mitochondrial dysfunction or not. And NIX is just there to clear out dysfunctional mitochondria from that stress. So these are just the Western blots that go along with this quantification. And we also said, hey, pacing induces quite a profound calcium signaling pathway. What's going on with calcium? And what we found in pacing, the amount of calcium that ends up in the mitochondria is really quite trivial. It didn't really accumulate there to any great extent. So we started using other calcium biosensors targeted to other organelles, and the nuclear localized calcium one was the one that lit up the most. And so we see a real pacing induce nuclear calcium accumulation, which is quantified here. And that kind of, to me, was really exciting, is what if NIX is contributing that? And in lipotoxicity, the calcium gets pushed to the mitochondria and activates DRP1. And in pacing, the calcium gets pushed to the nucleus and activates transcriptional regulators. So we really played with that idea for a while, and kind of supporting it, we didn't really see any changes in phospho-DRP1, which went to the mitochondria, and the calcium didn't go to the mitochondria. So we kind of played around with that idea for a while. We also looked at mitophagy, and under pace cells, we do see an activation of mitophagy. We isolated mitochondria, and we can see autophagy proteins like LC3 and P62 accumulating in isolated mitochondria from our stimulated myotubes. And then we started knocking down NIX, and you can see the mitophagy response from stemmed muscle was blunted when we knocked down NIX, and that's quantified here. And then when we turned our attention to the nuclear calcium accumulation, which is induced here by stimulation, again, knocking down NIX blunts that response. So we see NIX now as kind of a calcium regulator and a mitophagy regulator together, and bringing together lysosomal signaling pathways. And that's really where I started thinking about this term nexus a lot. So the next question was, if when the calcium goes to the mitochondria, we activate signaling pathways that are directed towards the mitochondria. If we put the calcium in the nucleus, are we going to activate signaling pathways and calcium regulated transcriptional regulators in that nucleus as well? And so we looked at NFAT-YFP, which was given to me by John McDermott's lab and cloned by Tetsaki Miyagi. We see that NIX could put NFAT-YFP into the nucleus, quantified here, and suggesting that that's where NFAT would be activated. And we also saw that the transcriptional repressor, HDAC5, which is normally in the nucleus, gets pulled out where its repressive effects would be inhibited. We didn't see the same effect with HDAC4, which suggests that there's some specificity here. So that project right now is being run by Jared Field, who's a PhD student in my lab, and he's looking at gene expression changes. But along the way, we also decided that, hey, now it's time to get back in vivo. And to do this and to really get in to dig deep, we decided to generate a muscle-specific NIX knockout mouse. We have a transgenic core at the University of Manitoba at our Bannatyne campus, and they were quite excited to jump into CRISPR-Cas9 technology. And using Cas9, the director of our core, Dr. Barb Triggs-Rain, inserted LOX-P sites around exon 2 of NIX. And then I purchased the human alpha skeletal actin driven CREMOS from Jackson Labs. And I chose this one to get the maximum specificity in skeletal muscle with avoiding the heart, because MCK Cre does have a big cardiac effect. And also this HSA1 has a DOX-inducible version, or excuse me, a tamoxifen-inducible version, which I thought might be handy down the road. And so when we bred these mice together, we essentially Cree takes NIX out and we get a muscle-specific knockout mouse. And when we look at our flox mice versus our knockout mice, we got pretty good knockdown in gastroc plantaris. We have no impact of NIX on the heart, which was really the goal in using this approach, and no impact on the liver. So Jared and I set about on the task of collecting as many tissues as possible and running as many histological tests that we could. The Department of Human Anatomy and Cell Science houses our histology core. So I hope that puts me to the front of the line, though I wouldn't want special treatment, but we do utilize this core quite a bit. And while we were looking at H&E staining of our flox mice and our knockout mice, Jared and I noticed that some of the myofibers just didn't look right. And that's hard to describe what we were seeing, but maybe it's slightly round. It just didn't look right. It looked like they were islands onto themselves within the sea of fibers. And so we tried staining with Gamori trichrome. And Gamori trichrome essentially is a clinical test to evaluate a ragged red fiber phenotype. And what we saw pretty universally, especially in our male knockout mice, is that every gastroc plantaris that we examined had several ragged red fibers, which is an accumulation of red staining. And ragged red fiber is a sign of mitochondrial myopathy. And typically the mitochondria in this myopathy are accumulating in the subsarcolemmal space, and then they stain red in the Gamori trichrome. We didn't see any in our flox mice, male or female, and literally every male knockout mice had evidence of these ragged red fibers. And since the literature told me that the mitochondria were accumulating subsarcolemmally, or in the SS space, we did EM. And when we looked at our flox mice, we had very normal looking mitochondria. We did wild type mice, we did Cree-only mice, and they all pretty much look like this. But then in our knockout mice, we saw something really quite remarkable. We saw accumulation of really large myopic mitochondria, mostly SS, but we do know that IMF, or the intramyofibular mitochondria, are also dysfunctional, but they're harder to assess because the myofibrils kind of compress them together. And in subsarcolemma, we can really see the dysfunctional mitochondria. We also saw vacuoles, which I've put stars on, and lots of them. And it's not really shown all that well here, but you can see there was granules. So vacuoles and granules and mitochondrial myopathy. And so Jared and I were just determined to figure out what is this and what does it resemble? And is there anything out there in the literature that can help us? And as we started investigating just through lit searches for this phenotype, we found this disease called Pompe disease, or some people call it Pompe's disease, some people call it Pompe disease. I'm going to go with Pompe. Pompe disease is a glycogen storage disease, and the granules that we're seeing are likely glycogen, and I'm going to show you some evidence that they are in a minute. But a glycogen storage disease, this is type 2 glycogen storage disease. This Pompe disease is also, it's categorized as a second disease, which is a lysosomal storage disease. So it fits into both categories because the cause of this disorder is a recessive genetic deficiency in lysosomal acid alpha glucosidase. And so this is a lysosomal defect. So the lysosomes don't become acidified. And so we got kind of excited about this because it suggested that if that's our phenotype when we take away Nix, that Nix is somehow at this nexus of impaired, or our Nix knockout mice have a phenotype where Nix is at the nexus of mitochondrial clearance and quality control, but also signaling pathways emanating from the lysosome. And as we looked a little deeper, patients with Pompe's disease have impaired mTOR signaling. And in fact, activating lysosomal mTOR with amino acid supplements can help alleviate a lot of the symptoms. And so this gave us a lot of ammunition to dig a little deeper and keep going. Now, these vacuoles we haven't quite solved yet. We think they're lipid containing. And we're still waiting on some histology to kind of confirm that. Oopsie. So what was next? Well, now, if our culture data could guide us, when we knock down Nix, we could make muscle more insulin sensitive. And so one of the first things we started doing was taking our flox mice and our knockout mice and doing insulin tolerance tests. And you can see that our knockout mice have a slightly greater sensitivity to injected insulin based on just a glucometer test. And so that suggested that our culture data was leading us in the right direction. We also tried to examine what are those granules. If those granules are glycogen, and this is resembling a glycogen storage disease, we should be able to stain for them using an acid shift stain, which detects polysaccharides. And then shown here, we can see that our flox mice have a real normal PAS staining, which is the acidic shift stain. But our knockout mice accumulate a much deeper purple stain, indicating that there's more polysaccharide in them, and in that context, it would likely be glycogen in muscle. And then lastly, we asked, are these mice going to be protected if we give them a high-fat and sucrose diet? And our culture data would predict yes. And so what we've done here, again, is an insulin tolerance test where I've taken the flox mice on a low-fat and sucrose diet, and this is a normal ITT response. When we put them on a high-fat and sucrose diet, they become insulin resistant. They don't respond to insulin in the same way. And when we put our knockout mice on a high-fat and sucrose diet, we have some partial protection, especially early in the ITT. Now, I should note this is very preliminary at this point. Our ends aren't big, but it's leading us, we think, in the right direction, and we're continuing to look at that. I also wrote a CFI grant along the way to try and bring metabolic caging and metabolic treadmills to the DREAM theme. And this would allow us to kind of pair, at least with the treadmills, what was happening in some of our human studies. The idea that we could collect VO2 and VCO2 in animals. We started with metabolic caging, and our hypothesis here was if our knockout mice have mitochondria accumulating that have become overloaded and are not fully functional, we could see a respiration impairment. And we see over the course of 24 hours, our knockout mice have slightly lower VO2s compared to the flox mice, and that's quantified here. The next step that we're taking with this is we're going to put them on the high-fat and sucrose diet, and in my hypothesis is we'll widen this difference when Nix would be induced. Next, Jared put his mice on the metabolic treadmill, and again, we saw kind of a subtle reduction in VO2 at every stage on the treadmill test, and that's quantified here. So without clearing the mitochondria out and turning them over in a quality control kind of way, we're inhibiting maximal oxygen consumption. The next piece of data I want to show you is new for us, and some people call it a kinomic analysis, but it's not truly the full kinome. So I'm going to try and refer to it here as a kinome peptide array. And we're working with a researcher named Jason Kindrachuk, who's developed this platform for us at U of M. And Jason Kindrachuk is actually quite famous in the pandemic because he's a CRC in novel viruses. So he's been busy in the last couple of years. But before things got too nuts, he helped us with this array that he had developed. And Jason was recruited to U of M to be part of our Ebola team. And that team, it works out of our level 4 microbiology lab in Winnipeg. And so they took essentially phospho acceptor sites from human, and they use a lot of chimps and macaques and rodents, and they created essentially a peptide kind of database for conserved phosphosites. And they have over 1,200 conserved phosphosites now in this database. And from there, they can generate peptides, which get mounted on a plate or a chip. When you take your sample and you mix them together and add a radiolabeled ATP, you can essentially get an array out of that, which is analyzed first for the kinase involved, or the known kinase involved, but then also through a software that puts bigger picture pathway analysis together. And so we've done some preliminary work with that, and we paired it with a gene expression array. And so before I show you that data, I want to just show you that we think we have a bit of a sex difference here, and we are evaluating it. And the reason we think that is because it was only males at 10 weeks of age of any genotype that had the ragged red fibers. So the ragged red fiber seems to be specific to males, at least at a younger age. So based on that, we decided if we're going to do this, we should probably look at males and females separately. And so this is our gene expression array. Green is the gene going down, red is the gene going up. And we looked at a bunch of muscle genes. And what we saw was quite interesting. The male mice did show some changes in gene expression in muscle genes, but the female mice showed a lot more. More genes were regulated and mostly down-regulated. Many of these genes are calcium regulated, and some of them are mTOR regulated. And so we think our males and female mice both have a phenotype, but it presents slightly differently in each. And then finally, we did this kinome analysis. And to do that, we treated mice with and without insulin for 15 minutes, both our flux and our knockout. And we saw really, really quite interesting things. First, we saw that insulin receptor signaling was upregulated in our knockout mice. And that was predicted based on our culture data. And concurrent with that, we also saw impaired or down-regulated mTOR-related signaling. So we feel quite confident that what we saw in culture is happening in vivo. We also saw down-regulation of signaling pathways involved in fatty acid and triglyceride production. And that might explain why those vacuoles are accumulating and as fat is kind of accumulating in the muscle. We also saw that was an upregulation. We also saw downregulation in calcium signaling, particularly with calcineurin and NFAT signaling in our knockout mice. And one thing we haven't actually gone after yet, which was a little bit of a surprise, is we saw a big downregulation in SMAD signaling, TGF-beta, and myostatin signaling. And this is kind of something that we haven't started going after yet, but it's on the kind of our future work list. So to me, these changes in signaling pathways really help confirm what we had in culture. I'm going to show you this last slide now, and it's very preliminary, but to me, it's really quite interesting. Everything I showed so far in the knockout mouse was really quite predictable based on our previous work. But science isn't predictable, nor should it be. If all our results were predictable, we wouldn't have to run experiments, and we do. And I always remember John McDermott telling me at one point in my training that about the burden of proof, and if all science was totally predictable, well, we wouldn't have to do what we do. And it's funny how some of those things stick with you throughout the course of your career. We started looking at not just insulin tolerance tests, but glucose tolerance tests. And this is where things in our knockout mice became a little bit unpredictable. And we had a hard time with this for a little while. When we took our flocks mice on the low-fat diet and treated them with glucose and watched their blood sugar, we see kind of a normal glucose response as it eventually gets cleared and returned to normal. Following high-fat and sucrose feeding, we see this exaggerated response where the blood sugar stays high longer. But when we looked at our knockout mice, even when they were on the low-fat and sucrose diet, they have an exaggerated response, indicating that they're a little bit more insulin resistant than the flox mice on this diet. And this was opposite to what we saw when the mice were on a standard chow diet. And it got us really thinking that the difference here really is the sucrose. And adding sucrose to the diet, which was really important to some of our models in gestational diabetes, is been very instructive, because it's showing us now that our mice with dysfunctional accumulated mitochondria, they're not able to handle sucrose in their diet. So these mice have some protection against the lipotoxicity when we remove NICs, but they have sensitivity to sugar. And that's really quite interesting. And now we're pursuing that. And so I have to thank Vern Dolinsky for really pushing us in this direction, because when he was developing his gestational diabetes models in rodents, just a high-fat diet was not enough to make them insulin resistant, the female mice insulin resistant, and to induce gestational diabetes. Sucrose had to be added to the diet in female mice to get that insulin resistance, and then ultimately GDM. And so now by looking at this, we kept the diet because it allows us to do more sex-specific investigations, but now it's also elicited that there's a difference in these knockout mice between chow and a sucrose-containing diet. And so this is where we're investigating right now. Like I said, this is very preliminary from Jared's work, but we're pretty excited to go after something that wasn't quite as predictable. Okay, I thank you very much for your time. I have to do my acknowledgements. The new work I showed today was a PhD student in my lab, Jared Field. The work that was published came from Simone de Silva Rosa, who has completed her PhD now, and she's a postdoc with Gary Sweeney. I've had a number of other wonderful students in the lab and RAs and techs who have really helped help us with this work. I also have lots of collaborators I really need to acknowledge. Vern Dolinsky was really instrumental early on at getting us into the high fat and sucrose versus low fat and sucrose diet, and looking at muscle insulin resistance in rodents, and providing us with tissues and whatever we needed. So I have to thank him. Barb Triggs-Rain generated our mouse and did such a great job. Grant Hatch is our cardiolipin expert and helps us with respiration. Saeed Gabami is an autophagy expert in all things, in all cells, and normal and cancer. So I have to thank him. And Adrian West helped us with the pacing. Michelle Aliani, I ran into at a conference in Banff, and we've been friends ever since, and he ran all our metabolomics. Aisha Salim is a new PI at CRIM, and is helping us with primary cultures, and we have a lot of future work coming together. And then Kinderchuk did our genomics during a pandemic when he was supposed to be studying COVID, and did it for authorship. So I have to thank him so much. These are our funders that have helped us along the way. I couldn't have done this without CRIM. They provide everything that we needed to do this work, and as well as some operating funding when times got tough. I'm big on Twitter, and I like to talk about results in science. I don't like to talk about politics. So if that's you, send me a tweet. And yeah, thank you very much. I'd love to take some questions.
Joe, thank you very much. What a wonderful story you've told us here about NICs, and you know, going from cell culture to animal models and the stories that parallel each other, and that's what you like to see. Sometimes twists in the end, but a great story, Joe. Thank you very much.
Thanks, Dave.
So the way the format is through Q&A, folks. Please ask your questions in the Q&A box, and I'll read them out for Joe and for everyone to hear, and I'm sure there's lots of questions coming up. Okay, from Anonymous, great talk. Are gender differences also observed in mitochondrial myopathy and Pompe disease? Do you know if there are? And you saw a change in VO2 in the knockout mice. Did you also see any changes in RER? Yeah, I don't know.
Okay, so lots there. I don't know about Pompe disease. I haven't dug that deep into it yet, but yes, our mitochondrial myopathy determined by ragged red fibers so far is exclusively in the male mice, though with the pandemic, we haven't been able to age mice much beyond 20 weeks. So I don't really have 1 year data or anything like that to assess an aging impact on the sexes. So I don't know if it's just it comes on earlier in males than in females, or if the females are completely spared from that. And the same answer applies to the gene expression array. We've only done that at around 10 weeks as well. I don't have kinome data yet for females. That is coming, but yeah, I'll just write that off to the pandemic and Jason getting busy having to work in a virology lab. What was the other one?
You saw a change in VO2 in the knockout mice. Did you also see a change in RER?
No change in RER yet. It is a reduction in both CO2 and O2. We're still kind of working out those details. The change is not, of a massive magnitude. So we're hoping that once they're on the high fat and sucrose diet for a while, that difference might get better. We think, or bigger, and we think that would support our hypothesis. And so far, males and females, even though our N's really low, don't seem too different in the cages or the treadmill.
Yeah, studying these sex differences has really revealed some interesting paths to take, hasn't it? I mean, it's really a worthwhile endeavor. So Dr. Hawk from McMaster says, great job, Joe. A small methodology question: when you were quantifying fluorescence in your cell culture, nuclear translocation, how did you quantify that? Was it per area or per myotube? Those are good questions to ask, because we often struggle with how to quantify these things.
Yeah, and we've done a number of different approaches. And actually, this is one of Jared Field's strengths. He tries 100 different ways to quantify everything. And we try and come to some sort of scientifically reasonable conclusion. We've tried tracing through the cells and looking at where the peaks are, kind of, do we get an M, do we get a peak? But I think in the data I showed you, it's a on-off kind of percentage of cells with the dominance of nuclear or cytosolic in that. And so that was kind of like the 3 independent experiment, 50 cells per condition averaged over the group. Okay.
Dr. McDermott says, nice job, Joe. You're a credit to us all. Curious about the connection pathway between palmitate exposure and NIX induction. Do you know anything about that pathway connection between the two?
Thanks, John, for the question. Yeah, there's a few things. Our work and Gerald Dorn's have shown that NIX expression is regulated by PKC signaling. Gerald Dorn showed it was directly through PKC alpha activating SP1 on the NIX promoter. And then in 2015, we showed that there was a second kind of indirect pathway where PKC delta would repress both MEF2 and SRF through phosphorylation, and that repressed a microRNA 133A, which we identified as a negative NIX regulator. So we kind of have inhibiting the inhibitor from our work, as well as direct activation of SP6 by PKCs. And so that's kind of our 2 pathways that are kind of our working models in palmitate exposure and NIX induction.
Okay. I guess I'll comment on the accumulation of large mitochondria when you knocked out NIX. That, I guess, is, sort of verification of knocking out mitophagy. I wondered about, it's not the only mitophagy pathway, of course, so the others are obviously not compensating to take care of this, pink park and that sort of thing. Have you measured any of those other pathways in mitophagy?
Simone, when in her work, did look at parkin, and parkin expression didn't really change much, and we didn't really see much of an recruitment to it, to the mitochondria, just by fluorescent imaging. And then I also had asked Simone to do a really extensive screen on just the muscle tissue of the low fat and high fat fed animals, and we used soleus muscle, and she looked at the expression of parkin, Reb, FunDC, FKBP30 or FKBP8, and BCL2L13, and none of the gene expression changes were as remarkable as Nick. So we also looked at BNIP3, and actually many of the mitophagy markers actually went down. The only one by whole tissue analysis that increased along with Nick's to a remarkable way was parkin. And so we do see it in induction of parkin on high fat feeding. So, but we never really went on further to see that, because in culture, Nix didn't seem to impact parkin and vice versa. So they're kind of two independent pathways, we think, that are going on. Though Åsa Gustafsson recently published that Nix and FunDC1 work together to induce mitophagy in cardiac progenitors, and so we're wondering if there's kind of a FunDC1 and Nix kind of cooperation, and that observation kind of really interested me, because BNIP3 is very hypoxia inducible in the heart and in muscle, but Nix isn't. And it's published that Nix is hypoxia inducible in lots of other cell types like reticulocytes and so on, but we never see Nix expression coming up, especially in the heart with hypoxia. And so I'm wondering if it's the interaction with FunDC, which is also hypoxia inducible, that kind of stabilizes Nix. And that kind of is supported by Osa's work. So yeah, so there's a bunch of things that we've thought about here in the other mitophagy regulators, and right now I'm thinking there's a complex, especially of the receptors. Pink and parkin seem to be quite independent of that and do their own thing. And what actually is stimulating pink activity in the first place, I think, is pretty interesting. I know CCCP is kind of the traditional way in culture to activate pink parkin, but that's not physiological. And Richard Ewell has suggested it's misfolded proteins in the mitochondrial matrix. So, and I'm not really sure how that would impact lipotoxicity, though misfolded proteins can happen, I think, in any form of mitochondrial stress. So that might be kind of the independent regulation in palmitate.
Makes a nice connection between the mitochondrial UPR and mitophagy regulation, maybe.
Yeah, so that's my Saeed Ghavami collaboration there. To everything UPR and other mitochondria or ER, and maybe now moving into an integrated stress response. So so that's kind of always on our hit list in our rhabdomyosarcoma projects.
See. One more question from me. You said something about a high-fat diet originally, right at the top of your talk, and that led to an increase in Nix and a change in cardiolipin maturation. I think you said something about. I wondered about the. Have I got that right? Is that correct?
We've never studied cardiolipin maturation directly, and it would be interesting to look at Grant Hatch's tafazin knockout mice to kind of go after that, but I know Grant has published that in the tafazin knockout mice they have insulin resistance, and he's got several papers kind of investigating how cardiolipin is involved in that. And as soon as I got that data from the metabolomics, I did run to Grant's office and showed him what was happening with all the different cardiolipin species. And he said to me that straight off that when you put animals on a high fat diet, you do see that 18-2, that alpha linolenic acid cardiolipin just deteriorate. And I don't think we know why. It might be just getting replaced by other cardiolipin species with other acyl chains.
Fascinating.
And on our shotgun metabolomics, I don't think we get I don't think we get the coverage really to look at really minor species, especially of cardiolipin. So we get the big one and we saw it deteriorate, but the other ones coming up were non-detectable in some samples. And so then wasn't really anything I could quantify.
Yeah, no, that's good. It's okay. That's interesting. Are there any other questions from the audience, please, through the Q&A panel? I'm seeing one more, just one moment here. John McDermott, if the NICS knockouts have increased glycogen levels, do they have a greater reliance on glycogenolysis and anaerobic metabolism? You should have to exercise them to figure that out, maybe, but yeah.
Yeah, we have blood, but we don't have lactate yet when they're coming off the treadmill. So that's a really good question. I don't have a really good answer. We just don't know yet. But that is one of the, like, blood lactate is on our list of analysis to do on the serum. And the trouble with mice is you just don't get a lot of it. So when you think about, oh, we want to do a dozen ELISAs and lactate, well, we have to move to a multiplexing model to get that. But in the future, hopefully, yes, because we do have a new multiplexing platform available.
Do they have the same exercise tolerance, the NICS knockouts and a wild type animal, or have you tried that?
I thought they were going to be falling off the treadmill in like 5 minutes, but it's not quite that extreme. It's we haven't done a total distance yet, and our protocol through animal care right now is just a quick graded exercise test where the speed it's like the Bruce protocol kind of idea where it increases every 2 to 3 minutes. So it's minor, but not significant yet. That's hopefully gets sorted out with more animals run.
Very good. All right, I think we've asked enough questions. We'll let Joe off the hook here on a Friday afternoon. Thanks everybody for joining this afternoon, and we'll see you at our next seminar in a couple of weeks' time. Thanks everybody, and thanks to Joe. Joe, hang on for a minute, please. And
sure. Thank you, everyone.
Seminar's over. Thank you.
Well, thanks again, Dave.
Perfect. Okay. Yeah, good. I was just getting rid of people there. No, hey, that was a great story. That was a very
Thank
good
you.
story. Yeah, no.
Thank you.
Nice. Yeah. Those NICS knockout mice are useful, aren't they? That's useful. They're showing you different pathways of calcium regulation and stuff like that. It's nice.
Yeah, we did the NKX Cree cross as well. So we have muscle and heart.
Oh.
That was my heart and stroke grant, but then they didn't fund it, but I think they ran out of money in the last competition. So I had to resubmit it.
Oh, I see. Just now in September, you resubmitted, or?
Yeah. I was pretty close, I think, but I think heart and stroke just didn't have the funds. So when I thank CRIM for bridging me, that's what they're bridging me for, is a lost heart and stroke grant. So.
Yeah. You know, I've had a heart and stroke grant in the past, and I find them such a high maintenance organization. I really, progress reports and all that stuff. I just, I don't know. I haven't had good responses from them lately, and I don't know why. Some of the reviews I get are just And I've served on the panel once anyway, at least a couple, maybe times. And I don't know. I'm frustrated with heart and stroke.
Yeah.
I haven't gone back for a few years.
I feel you. I find I'm frustrated with actually all the cardiac panels right now. The reviews are just horrible. And it just seems like people are either funding what they perceive to be as famous scientists or their famous friends scientists, and it just anybody else are just knocking you down.
Yeah.
Even my brother used to have stroke funding through them, and he had a brain blood flow project, and submitted, just got triaged, and then he published the paper in Nature Communications. So it was just like silly, like yeah. So same thing. Like I have a BNIP3 project as well in the heart, which is probably dangerous for me, but it's very neonatal cardiology. So it's totally different model systems and that. And I have never had a grant with such strong data, because we had everything that we could think of thrown at it, just trashed so badly in the last 2 rounds of CIHR with really not much constructive to say. And people have even commented that I'm not studying a disease. And so this is a neonatal asphyxia hypoxia model, and we got all these clinician scientists lined up with us, from people that study congenital heart defects to diaphragm and lung defects to asphyxiation in a newborn to so there's a million different things that kind of feed into this hypoxic model, and just like, yeah, just crazy. And then you address them, you go to the stats. We're going to WHO, we were going to anywhere we get data on how many babies die of hypoxia a year around the world. And the numbers are staggering. And then to get comments like Dr. Gordon's not really studying any sort of disease, it's just kind of all like an academic kind of endeavor in hypoxia. And I'm like, well, okay, thanks.
Yeah. In these rooms, you need a champion in the room to sort of say, counter those kinds of comments that don't make any sense, or a chair that is actively participating. But being a chair and being able to make comments that counteract that is so much work that anyway, the review process is far from ideal, that's for sure.
The grant that gets our best reviews is this one on the muscle nix. So that's why I keep pushing it and keep saying we keep working on it. The pandemic hasn't slowed us down that much. We're making progress, and hopefully it gets funded at some point at a good level. And our comments have been good in the past. I think people are interested in the idea. And it's not my idea. Signaling pathways from the lysosome, I can't take credit for, but I think it's so cool. It's one of the reasons that it got me into autophagy and mitophagy in the first place, that I don't really care about recycling proteins and things like that. But as you're doing it, if you're launching signals out that are, dictating cell fate, I think that's really, really quite cool.
Yeah, I agree. Well, Joe, I'm going to let you go now and enjoy the rest of your afternoon. And I hope you're in, I hope things are going to work out well for you there. You definitely need that relaxation time in PEI. So hope you get back there soon. How long are you going to be in where you are now, Waterloo?
Just until Sunday morning. I'm catching a very early flight Sunday morning and back. So it's just like a 4-day visit, see the folks and my brother. When we started doing the math, it had been 3 years. So time flies.
Yeah. That's good. Nice reunion for you. That's good. That's good.
Yeah.
Okay. I'll say goodbye. And thanks again for doing this, Joe. Really appreciate it.
I appreciate the invite. Have a good weekend.
Okay.
Talk to you soon.
Yep.
Okay.
Yep. Bye.
See you, Dave.
Yep.